All right, hello everyone. Thank you for your patience. My name is Nancy Chen. I'm an assistant professor at the University of Rochester. And today I'm going to tell you about a project that we actually started during my PhD, and this is work done in collaboration with Elissa Sorojsrisom and Andy Clark. So I am going to try to shorten things, so if at any point anything is unclear, please do feel free to ask me during the questions or interrupt me. All right, so as we now know, there's considerable genetic variance for fitness in many different natural populations. Here I'm showing you kind of estimates of additive genetic variance in fitness for several different mammal populations, estimated from Bayesian animal models. And this has been long thought to be a conundrum, right, in evolutionary biology. Selection should act to remove variation over time, so why do we see so much genetic variation for fitness and fitness-related traits? There's been a ton of theoretical and empirical work over the past, many years focused on trying to understand what maintains genetic variation for fitness. There have traditionally been kind of two evolutionary hypotheses that have been invoked, right? So mutation selection balance or balancing selection. So according to mutation selection balance, right, mutation introduces deleterious variance to a population over time, which is then removed by selection. Whereas under balancing selection, here the hypothesis is that natural selection is primarily responsible for maintaining genetic variation in fitness. Balancing selection can take many different forms, right? So there could be heterozygote advantage, negative frequency dependent selection, or antagonistic pleiotropy or genetic trade-offs, right, which is the focus of this symposium. There are many different forms of antagonistic pleiotropy, right? So there could be life history trade-offs. A classic life history trade-off is a trade-off between survival and reproduction. There can also be trade-offs between early and late survival, and also sexual conflict or opposing fitness in males and females within a population. So traditionally, right, theory has predicted that there are actually only a very restrictive set of conditions under which you would expect for antagonistic pleiotropy to maintain genetic variation in natural populations. But more recent empirical work and also recent theoretical studies have actually shown that we can get maintenance of variation in fitness under antagonistic pleiotropy under a wider sort of conditions, especially when you think about the fact that there may be both kind of sexual conflict and trade-offs between different life stages happening simultaneously within a population. To date, there have been like a number of just studies that have successfully identified antagonistically selected loci. All of the toxins associated with the symposium are great examples. Here I'm just showing you kind of two textbook examples. So there's a locus that controls horn morphology, and so a sheep that's maintained by opposing kind of natural selection and sexual selection. And there's also a large effect locus in salmon that controls age at maturity with opposing effects in females and males. However, overall, it's still relatively challenging to identify antagonistically selected loci in nature. And there's also a need for more studies that kind of try to test for all sorts of possible trade-offs, right? So considering a lot of studies focus either on life history trade-offs or sexual conflict, and there aren't that many. There are a handful, but there need to be more studies that kind of look for signatures of sexual conflict and life history trade-offs in the same population simultaneously. This is really hard to do, right? Especially in natural populations, because that involves fairly detailed demographic monitoring and also the ability to genotype and phenotype large numbers of individuals. And in my lab, the way we get around this is by applying genomics to a handful of these long-term demographic studies where scientists have monitored a single population of individually marked individuals for decades. So the primary study system in my lab is the Florida scrub jay, this really beautiful bluebird here. It's a cooperative breeder, so offspring delay dispersal to stay home and help their parents raise future offspring. They're restricted to this really unique fire-maintained scrub habitat in Florida. There are a couple aspects about its biology that make it particularly amenable to long-term demographic studies. So they're non-migratory, highly territorial, and philopatric, so individuals stay in the same area throughout their lifespans, and their offspring don't move very far, so you can track individuals across multiple lineages across multiple generations. They're socially and they're mostly genetically monogamous, which means that we can construct fairly accurate pedigrees from field observations alone. For the field biologists out there, you also appreciate these birds are addicted to peanuts, which makes them really easy to work with in nature. But I should say that we try our best to not interact with these birds too much. So there's been a long-term demographic study of Florida scrub jays at Archwell Biological Station that started in 1969. All the birds in our population are uniquely banded, so we can identify them in the field. There's a ton of work that goes into the study. So the entire population is censused once a month, providing really detailed records of individual lifespans. Every nest of every family group that we follow is found and carefully tracked, so we know exactly how many eggs every pair lays, how many of those eggs hatch, and the fates of all of the nestlings. We have spatial records. We know where individuals are located in space over time, which allows us to measure dispersal distances. Importantly, for genetic and genomic work, my collaborators started taking blood samples from every single new nestling or immigrant that arrives in our population over time, starting in 1999. So we have this really nice archive of DNA samples of all the individuals in our population over time. We also have measures of a whole suite of relevant ecological data, so information on habitat quality and composition, climate variables, fire history, food abundance, et cetera. And so what this means is that over the past half century, we've accumulated complete and really detailed life history records for more than 14,000 individuals on a multi-generational pedigree. I'm just showing you our population pedigree here. All you need to know is that we have a lot of data. And then, importantly, now we've overlaid on genomics. So as a grad student, I designed and sequenced and assembled the genome, designed a custom Illumina B chip, and we genotyped about 4,000 individuals at 10,000 SNPs across the genome. This plot on the bottom shows the total number of individuals in our population over time in gray, and the blue indicates individuals who are genotyped. So we've genotyped nearly every single individual in our population for more than 16 years. And so this kind of wealth of genomic data, demographic data, and phenotypic data on a population with a known genealogy gives us a lot of power to kind of test evolutionary predictions in a wild population. So from our demographic data, we notice that there's really high variance in lifetime reproductive success. Here, I'm defining lifetime reproductive success as the total number of offspring produced by an individual over its lifetime. And here's data for about 1,000 dead birds. You can see most of them do not produce any offspring during their life, but some actually produce more than 40. So we used McMick-Glim to fit animal models to estimate kind of additive genetic variance for lifetime reproductive success. We did this for 2 sets of individuals. So LRS all means for every single bird born in our data set, so every tracked individual. And then we also looked at individuals who actually successfully became breeding individuals. And if you focus on the purple histogram, that shows additive genetic variance, and the asterisks are showing significant variance components. And so we find that lifetime reproductive success is heritable in females, but not in males. Now, as you all know, fitness is a really complex and composite trait and has many, many different fitness components. So this idea was first kind of brought up by Timothy Prout in the 60s. And a lot of this project was kind of inspired by this paper by Christensen and Friedenberg on selection component analysis, in which they developed this really hierarchical series of tests to study kind of selection acting on different life stages, different stages of the life cycle separately. So here I'm showing you kind of a typical life cycle diagram for a Florascribia or a sexually reproducing diploid organism. So there can be differential survival, right, of zygotes to adults, which allows for viability selection, et cetera. Not every individual who survives to become an adult successfully breeds, so there are opportunities for sexual selection. Individuals who do breed may have differences in fecundity, right? And there may also be opportunities for segregation or gametic selection. We did test for segregation distortion, and we found one region of the genome that seemed like it was being transmitted unequally, but it's near an indel. We did like whole genome sequencing data, so we're not super confident. So I'm not gonna talk about segregation distortion. We looked at 3 fecundity measures, so number of eggs laid in the first clutch of the season, number of hatchlings, and then annual reproductive success. Florascribes can delay dispersal, so kind of testing for sexual selection was a little tricky. The best measure we came up with was for all individuals who are 2 years old, are you a breeder or not? And then we also considered many different stages for survival. So here's a typical life of a Florascribia. We band individuals when they're 11 days old, so that's when we can start tracking them. Scrubjays typically stay in the nest until day 18, after which they fledge and kind of hang out as pretend to be little pine cones until day 30, after which they're a little bit more mobile. By day 90, they're nutritionally independent from their parents. By day 300, they're physiologically capable of breeding. And then we also see whether or not they actually establish as a breeder, and then their breeder lifespan. So we considered a whole suite of different fitness components. We ran animal models to do variance components analysis and estimate kind of how much additive genetic variation there is for these different fitness components. I know this is a lot of plots, but the key messages here are natal nest explains a lot of variation of survival from day 11 to day 30, which makes total sense, right, because they're still in the nest for half that period. And then we find heritability of female survival from day 30 to day 90, but not males, and then male egg count. All right, so I mentioned earlier that we have genomic data for a bunch of individuals, so then we set out to do genome-wide association studies. We did perform genome-wide association studies of lifetime reproductive success, in addition to the whole suite of different fitness components that I mentioned earlier. We included the kinship, so the genomic relatedness matrix, to control for relatedness in our sample, natal year and natal nest as random effects. We also recognize that there are many environmental factors that may influence individual variation in fitness. And so we tried to account for that because we had a lot of measured variables. So we considered attributes of the individual, including inbreeding coefficient, whether or not they're an immigrant. We had a bunch of attributes of their natal territory, their natal environment, right? So number of helpers, territory size, habitat composition, age and experience of their parents. And then we had a whole suite of environmental factors, so population density, rainfall, temperature, food abundance, et cetera. So for each of our fitness components, we first performed a variable selection step where we determined which of these many variables actually had a significant association with variation in that fitness component, so we can control for those in our genome-wide association studies. And then we would fit the genotype of all of our SNPs across the genome and test for significant genotype-phenotype associations. We did so we ran additive models, but we also coded genotypes assuming the major or minor allele was dominant, so we ran a lot of models. I'm just gonna show you a few highlights. So for survival, here's just the raw data. We fit logistic models asking whether or not a given individual survived from one of these milestones to another. We're interested in looking at sexual conflict, right? So we fit separate models for males and females. And surprisingly, we did find two major effect loci that are associated with female survival from day 11 to day 30. So for this particular interval, the fixed effects that we needed to include in our model were incubation date, hatch number, inbreeding coefficient, drought index, and maximum temperature during this time. We found one allele on chromosome 11 and one on chromosome 15 that had pretty large effects on survival. So for instance, here's the predicted values for the SNP on chromosome 14, and you can see that females with like the opposing homozygotes have a difference in survival probabilities of like 0.3, which is quite large. For fecundity, I'll just show you in some data for egg count. So this is the number of eggs laid in the first clutch of a season. Distribution's a little wonky. Pretty much almost every pair lays 3 or 4 eggs in a nest. Interestingly enough, the fixed effects that were important for males and females were slightly different. So nest date was important for both, and the relatedness of the pair was important for both males and females. But for female egg count, the number of immigrants in the pair, number of helpers, and the experience of her mate and fire history of her territory also mattered. We found 3 alleles in females that were significantly associated and 2 totally different SNPs that were significantly associated with variation in egg count in males. So overall, across all of our different fitness components, we found 18 kind of large effect loci that are associated with variation in survival and fecundity. Most of them are in females and not in males. So here I'm just showing you effect size estimates for 3 different genotype models, and the significant hits are highlighted in colors. Now, we did estimate effect sizes for many different fitness components, and so one of our first questions was how, like for a given SNP, are SNPs associated with survival? Are they also associated with reproduction? And so here I'm showing you the minus log 10 p-values for our kind of 18 large effect fitness loci, I guess, are shown in columns. And you can see the p-value for their effect on all of the different fitness components in males and females separately. And I'll just tell you, it's a little hard to tell from just looking at this graph, but essentially every single one of our hit loci has a suggestive negative effect in some other fitness component. And we need to do a little bit more work and a little bit more simulations that show that kind of these opposing effects are strong enough to overcome drift and maybe explaining why we see kind of variation at these particular SNPs. But of course, fitness is a super complex trait, right? We expect it to be highly polygenic. And so it would be perhaps more interesting to take a more polygenic approach to looking for trade-offs. And so what we did was we estimated correlations between effect sizes for our SNPs genome-wide. Well, not really genome-wide. We ran it for all SNPs, and then we also only considered SNPs with a raw p-value less than 0.1. And so I'm going to show you these significant correlations, and just to orient you, first I'll show you female, the responses for female, and you can see lifetime reproductive success as well as all of our different fitness components. These are the correlations and effect sizes kind of essentially genome-wide between all of our different metrics of fitness. For males, it looks similar, and I just wanted to highlight a couple patterns. So if you look at correlations with lifetime reproductive success, you'll see that all the fitness components are positively correlated with lifetime reproductive success, which is good. And the strongest correlations are with kind of whether or not you survive to become a breeder for all individuals, or for our group of individuals who were breeders, what matters most is their lifespan as a breeder. So this is consistent with life history theory, where for longer lived individuals, survival is going to be the primary determinant of variation in lifetime reproductive success. We also see some evidence of trade-offs between survival and reproduction, right? So for instance, female breeder lifespan alleles that are associated with longer breeder lifespan are negatively associated with egg number. And then kind of early life survival in males is negatively associated with all of our metrics of fecundity. And we also see some evidence of trade-offs between early and late survival within sexes, right, where some alleles that confer increased survival during an earlier time period are associated with lower survival during a later time period. And then finally, if we look at kind of correlations between effect sizes between females and males, you can see that there's quite a bit of evidence for sexual conflict. One thing to note is the diagonal of the square would show correlations between the same fitness component between the sexes, and you'll notice that those tend to have positive correlations. We're really only seeing sexual conflict when we're comparing different fitness components between males and females. Okay, so to summarize, and I apologize again for kind of rushing through this, but we performed a very detailed analysis of lifetime reproductive success and of many different components of fitness in the wild population of Florida scrub jays. We found that lifetime reproductive success is heritable, as well as a few other fitness components, and lifetime reproductive success is primarily driven by variation in survival. We did find a small number of kind of major effect loci that are associated with variation in survival or fecundity, and many of these have opposing effects in other fitness components, and we're doing more analyses now just to kind of make sure we can definitively prove that these different opposing effects are what's underlying kind of maintenance of genetic variation. And then finally, if we take a genome-wide approach, we can see evidence of trade-offs between survival and reproduction, early and late survival, as well as sexual conflict across the genome. So none of this work would be possible without the many, many amazing students, staff, and interns at Archibald Biological Station that have collected the data over the past 50 plus years. I'd like to thank my collaborators, my wonderful team, and my funding sources. Also, I wanted to make a quick plug. I have 2 grad students and a postdoc here, and they're presenting Monday and Tuesday, so please go check out their talks. And if there's time, I'd be happy to take questions. Thank you so much.
Thanks, great talk. I think we do have time for a question or two. Yes, Linda?
Were you surprised by the rather limited extent of sexual antagonism that you found?
Were we surprised by the rather limited extent of sexual conflict that we found? Not too surprised, I think. So there's very little very little differences between the sexes in like survival and reproduction. They're genetically monogamous. So we didn't, like in terms of demographic processes, the only real sex bias that we observe in our population is in dispersal. So I guess it wasn't we weren't that surprised.
Yes.
Really stuff. Have you looked at the distribution of the loci that were associated with fitness traits over time? Did you have this pedigree?
Yeah, have we looked at the distribution of the loci associated with fitness over time? Yeah, so if by distribution of loci, did you mean allele frequencies? Yeah, so we have, so we've done previous work looking at allele frequency trajectories of all of our SNPs. None of these were associated with significant allele frequency shifts in our population over time. I wanted to show the trajectories, but also realized we needed to do a little bit more work to show that it's actually selection and not drift. Yeah.
Thanks everyone for coming, and to Carl and Luke for inviting me to come here. So I'm going to talk a bit today about the work we've been doing recently on genetic polymorphism in sexually antagonistic traits. Given this is like the 6th talk on sexual antagonism, I'll go quite quickly through this start. But males and females maximize their fitness in very different ways, and this can lead to a conflict known as sexual antagonism, because many of the phenotypes that are underlying male and female fitness will have a shared genetic basis. And so this means that genes encoding these traits can have antagonistic fitness effects. So they're beneficial in one sex when expressed in one sex and deleterious when expressed in the other. And this is known as sexual antagonism or intralocus conflict. So sexual antagonism displaces both sexes from their respective fitness optima. And we call this the sex load. So sex load has a number of important eco-evolutionary implications. Namely, it constrains adaptation. And so this has demographic implications like extinction risk. And what I'm going to be talking today about is the fact that a number of authors have highlighted that this antagonism may drive patterns of genetic variation. So this intuition has mainly come from single locus population genetic models. So these models consider one locus where one allele is favored in one sex and the other allele is favored in the other. So here, big A, you see my mouse? Big A is favored in females and small a favored in males. And so something that has come out of these models is the role of dominance reversal, at least for autosomal genes, in leading to balancing selection and driving elevated levels of genetic polymorphism. And the intuition for this is that if I have saturating fitness landscapes, so this is the dark red curve and the light blue curve. If these fitness landscapes are saturating, then actually the sex average fitness peaks in the middle, and that's the phenotype that's encoded by heterozygote, this big A, small a. And so this, even with additive phenotypic effects, we end up with dominance reversal for fitness, where on average across the sexes, the heterozygote is the fittest genotype. And there are other dominance-based arguments for sex chromosomes, but this is the intuition why balancing selection may be quite a pervasive feature of sexual conflict. And so kind of just to prove to you that this works, I ran a simulation of one of these loci where I have one allele that adds, one allele A that adds an amount onto a trait value and another allele that takes an amount off a trait value. Females want to be bigger, males want to be smaller, so it's something that looks like this. And what I plotted here is the heterozygosity through time. I ran it with really strong selection because I was impatient, so that's why this is so stark. But heterozygosity, I started at an intermediate level. I started the allele frequency at intermediate level, and very quickly goes to a very high level of heterozygosity that's maintained through time. But the question is, what about quantitative traits, particularly in particular polygenic traits? Because many of the traits that have been looked at in quantitative genetics are probably polygenic, like human height, or this is wing length in a migratory bird, and these are some of many studies of sexual antagonism there that really are looking at quantitative traits. So do we have any intuition on how polymorphism should be maintained in these kind of traits? Well, yes. People have suggested that dominance reversal, this pattern that we saw before, for fitness should occur across multiple loci. And partly because it's a really nice paper and partly because he's here to defend himself, I got a quote from Carl saying the average locus underlying two polygenic homologous phenotypes with antagonistic fitness effects is expected to exhibit partial dominance reversal for fitness, assuming that you have these saturating concave landscapes. And we wanted to test this, basically. Is this really the case? So to do this, I'm going to show you some simulations of a polygenic trait with L additive loci. At the kth, so each locus, one allele adds something on, and the other allele takes something off, and we're going to assume this amount is the same for all the loci. So it really is a situation like this where one sex wants all big A, the other one wants all small a, but we have a continual range of genotypes in between. So I'm just going to show you show you how there's an illustrative example, some simulation results. So it's the one locus case we saw before with these fitness landscapes. But then if I move to two loci, I see completely the opposite. Again, I've assumed really strong selection just for illustrative purposes, and that's why it's such an extreme pattern. But with 4 loci and with 10 loci, you see that we are losing heterozygosity through time. So the same fitness landscapes are now producing the opposite pattern of in terms of genetic variation. So to investigate this and why this was occurring, we analyzed a model of the fitness, we characterized the fitness landscapes and did analysis of gene fitness in order to infer actually what is the nature of selection that arises from these landscapes. And the answer actually is that when we have these saturating landscapes like this, what we have, even though the sexes are pulling in different directions, actually what you have is stabilizing selection. When you average across the sexes, selection is pushing towards an intermediate value. And we know that stabilizing selection purges genetic variation. So across a quantitative trait, polygenic trait, we are going to lose genetic variation. And what happens under stabilizing selection is that you can maintain, for any number of loci, you can maintain a maximum of one that's polymorphic. And this happens when your equilibrium phenotype in the middle is awkward in that it sits between two homozygotes. And so you fix all the loci except one, and then this last one just flips. And that's actually what the single locus models have been doing, is they've been assuming, they've kind of been getting rid of all the other loci and looking at this like one kind of remainder locus and looking at, okay, when do you maintain 0 or 1 polymorphic locus? We can only maintain a single polymorphic locus. This is a known result from stabilizing selection. So now we know that these saturating landscapes are not conducive to polymorphism. We said, okay, well, are there any other ones that can do it? And we found this is the condition for polymorphism to be maintained across loci for arbitrary male and female fitness landscapes. Prime here refers to differentiation. So I'll take you through what this says, which is that fitness landscapes must be first accelerating around this intermediate point, and they must be very steep, so very strong. And so this generates frequency-dependent selection that pushes you towards an intermediate trait value in between the male and female optima, at which point the curves are accelerating around, and this leads to diversifying or negative frequency-dependent disruptive selection. The tension between these two forces, in that you're pushing towards a fitness valley, and this generates elevated genetic variation across loci. But the condition, and to show you, this is a simulation with curves that look a bit more like this, and you can see we generate polygenic polymorphism. The issue here, so firstly, in terms of like biological plausibility of this kind of landscape, I mean, it's strong selection. We don't like that. We tend to think that that's not the case. And accelerating landscapes, and this is not what we think most landscapes probably look like. But actually a bigger issue is because you're driving the population towards a fitness valley, the sex load is extremely high here. So anywhere where you have conditions for diversifying selection, you're going to have extremely strong selection on any kind of modifiers that alleviate sexual conflict, any kind of gene expression modifiers. So this means the conflict is not really going to persist. So for instance, these are just some simulation results of Gaussian and power functions, and on the x-axis we have the heterozygosity. Actually, you can't see all of it, but what the dotted line that you can see, the vertical one, this is the, well, actually it's been difficult without. What you'll take from this is that the sex load is extremely strong. To cause a doubling in the level of heterozygosity, you need at least a sex load of 20% for power functions and 40% for Gaussian functions. So this is really, really strong. And we also looked at the sex chromosomes. So on the X, we found that, again, you need diversifying selection. You need negative frequency-dependent disruptive selection for polygenic polymorphism. And the conditions actually for this on the X are more restrictive than on the autosomes. The intuition for this is the fact that males send all their X chromosomes into their daughters, and females, mothers do half into sons and daughters. And so this means that if a gene is beneficial in males, this gene becomes overrepresented in the male gamete pool due to selection, so it's disproportionately pumped into females in the next generation. So for a given allele frequency, if you look at the frequency of this gene in males and females, the male beneficial ones are slightly more in females, and the female beneficial ones are slightly more in males, and this opposes the maintenance of variation. You have this like negative correlation between the context in which genes are good in and whether they're actually found there. On the pseudo-autosomal regions, you can get actually the opposite because of linkage with a sex-determining region. This means that the male beneficial gene becomes associated with a male sex-determining region, for instance, and then the female beneficial gene become associated with the X, and this leads to a positive correlation between context and whether or not these genes are found there. Okay, so to conclude, we integrated the phenotype into models of sexual antagonism. We found that it's probably a poor driver of polygenic variation because the conditions are restricted and they're really associated with a high sex load, so you won't really see this conflict persisting. And actually had a look through some databases of sex-specific selection gradients that have been estimated from wild and populations of humans, and we found that none of them satisfied our criteria for diversifying selection. And so, yeah, this lack of polymorphism may be an issue for empirical tests of sex-specific variation. And I should thank Charles, Max, Vincent, and Sally, the theory group in Lausanne. And if anyone is interested, we've had a preprint up for about a year now. So if anyone's interested in reading, thank you.
Thanks, Ewan. Great talk. Plenty of time for questions. Yeah, Nick.
Hi. Yeah, great stuff. Two questions. Surely this would be worse in a smaller population. Population size has got to influence the speed at which variation is lost.
In the sense that obviously drift will oppose any effects of selection. And here, because selection is stabilizing, so it's purging variation, so actually drift should increase the segregation time.
Yeah. Okay. And then the other question is, surely if there's incomplete penetrance of the dominance in biological situations, dominance is never perfect. So we're sort of in a drift weak world here, right? I mean, in terms of purging. So wouldn't that maintain if dominance wasn't perfect? Wouldn't that hang on to?
So here phenotypically, we have no dominance. All the dominance comes from, in this single locus models, the dominance comes from the fitness landscape, this non-additivity in the landscape, rather than dominance on the phenotype.
Yeah.
So I've never considered non-additive phenotypic effects. So it's not really phenotypic dominance. Drift really. I think drift will aid the maintenance of variation here because we're generating stabilizing selection. But yeah, I'm sorry, I'm not answering. We can maybe talk later. But.
The dominance is the residual variation off of the expectation, right? So of the intermediate genotype.
Yeah. Yeah. Okay. Yeah.
Get some sort of form of relaxation, and that might.
Okay. Maybe talk later. Yeah. Sorry, I'm not.
Is it quick?
Yeah, so you were talking about the needed this very strong selection, like 20% is at some point. But then this will be divided over very many loci because you consider many loci. So will it be that strong per loci? I don't know if you would have like 50 loci and they all affect body size. So maybe body size will have a selection 20%. Is that definitely, is it unreasonable high to have 20% for a trait part?
I mean, I guess the question is, yeah. Modifiers that could affect multiple loci will obviously be under strong selection. I mean, it may be true that individual cis modifiers of individual loci may not be under super strong selection, but any kind of genetic mechanism that can mediate the conflict, will be under strong selection. But it's true there may be a drift barrier but drift may be strong on individual small cis modifiers. Yeah.
Okay, Mattias, you're cutting into your own time. So let's thank Ewan again. Thank you.
Hello everyone, my name is Mattias Siljastam, and I'm gonna talk about a mathematical model about the evolution of allelic diversity and the sex-specific dominance at a sexual antagonistic locus. So it shares a lot of similarities with the previous talks, but it includes a new aspect, which is the sex-specific dominance, which will produce some strikingly different results. So, as we all know by now in this symposium, that many species are sexually dimorphic and with males and females with different phenotypic optima, which can generate sexually antagonistic selection. This can, in theory, maintain genetic diversity, and current theory suggests that at a single locus that we can get two coexisting alleles under sexually antagonistic selection, but this will require some rather strong and symmetric selection, so it's like a rather narrow parameter space where we would get this, as was pointed out in the previous talk. But when we look in nature at examples of sexually antagonistic genes, we find those that are highly polymorphic. So, for example, seminal fluid proteins can have over 20 alleles at a single locus, suggesting that there are a potential discrepancy between theoretical predictions and observations. So, here is a classical example of locus under sexually antagonistic selection, where blue gives fitness for females and orange fitness for males. And we can have something that is called sex-specific dominance. And importantly, in this talk, I consider dominance not specifically at the level of fitness, but on the level of the trait. And that in turn can result in this kind of dominance reversal in the level of fitness, where a heterozygote will express the gene in a biased way towards the one that is better for its own sex. So if these two genes, that one is beneficial for male and one for female, then a heterozygote will have a biased expression for the male beneficial allele if it's a male, or a female beneficial allele if it's female. So this will alleviate the conflict, at least for the heterozygotes, but the conflict still remains for the homozygotes, which will, if they end up in the wrong sex, then they will have a low fitness. So there are examples. Question like, does sex-specific dominance occur in nature? So a classical example by now is this salmon experiment, where they found this major effect loci affecting age of maturation, which has different optima for males and females, which were clearly was having a sex-specific dominance. We also have a paper by Grayshop and Arnquist looking at a large number of genes in seed beetles, showing that sex-specific dominance might be common, surprisingly common among genes overall. And the current theory suggests that sex-specific dominance will increase the parameter range where we can get this maintenance of allelic polymorphism. However, these models are mainly these classical population genetics models, one loci, two alleles, one locus, two alleles, and are kind of blind whether this mechanism can maintain more than two alleles. So, to my model, so I will today focus on the part of the model where I model an autosomal locus. So this autosomal locus is affects a quantitative trait that is under different optima for males and females. And so here we have survival on the y-axis and phenotype on the x-axis, where with female in blue and male fitness in orange as previously. So they have different phenotypic optima. And then by using Gaussian functions, we make sure that the function behaves smoothly also around the optima, where wide Gaussians result in weak selection, and more narrow Gaussians result in stronger selection for this trait. And then we also allowed for a mixed selection between the traits. So this phenotype is then determined by the two alleles at the locus. One can assume just additivity, where the realized phenotype is just the arithmetic mean between what the phenotype coded for the two alleles at the loci. Here is just how such a heat map where we have the allelic values on the x and y axis and the phenotype in the middle. It looks like this when it's just purely additive. But then I want to incorporate dominance. And when you have more than two alleles, or spectrumal alleles, then it's not actually trivial to incorporate dominance. In this model, I assume a dominance hierarchy, meaning I'm using constructed this function where I have a dominance parameter d. A positive dominance parameter means that it's always the allele coding for the larger phenotypic trait that is partial dominant. Or if we have a negative dominance parameter, then it's always the allele that codes for a lower trait that is partially dominant. And in the extreme of having very large d or very large negative d, we approach the max function or the mean function, meaning that we have a full dominance always of the one being larger or smaller. And then by allowing this dominance parameter d to be sex-specific, we get sex-specific dominance in this model. To analyze the evolutionary behavior of this model, I use the evolutionary invasion analysis with the adaptive dynamics framework, which relies on a set of assumptions. It's robust to deviate a little bit from this, but so the one assumption is that you have a large population such that the dynamics are close to deterministic. And then we want to assume infrequent mutations. So here we have the allelic trait value with the female optima and the male optima. And then we have evolutionary time. So here, now the orange here, we have a mutant introduced in the population. So with rare mutations, there's only a single mutant at any given time. And with small mutational step size, we can show if the mutant can invade, then it will actually replace the resident strategy. And we will get a trait substitution sequence where evolution will gradually evolve in the direction of the selection gradient until the selection gradient becomes 0. Then we can even be at a fitness maximum point, and then it's just an evolutionary stable point. So then, in this case, we would have an intermediate, an allele coding for an intermediate trait that takes over the population. And here is an actual individual-based simulation where we once again have allelic trait value on the x-axis, and we have evolutionary time. So here we just have one allele in the end. But it can also turn out to become a fitness minimum once reached, and this is what we call in adaptive dynamics, we call this, it's a branching point, and that's a point where genetic polymorphism emerge. And here is an example of an individual-based simulation where we in the end have two coexisting alleles, one that is specialized towards the female optima, and one is specialized towards the male optima. So the results when I analyzed my model was that this initially evolution will lead towards an intermediate allele that maximizes geometric mean fitness across the sexes. But once this is reached, then if selection is weak or asymmetric, this will be the endpoint of evolution. It's only when it's strong and symmetric that we will get this branching behavior. However, I also allow for this sex-specific dominance, and with that, we find that the condition for branching becomes much easier, so that we get polymorphism for a much larger part of the parameter space. And this is the branching condition, but yes, let's move on. So just reviewing some really old results. Here we have strength of selection. This is from the 70s, Kidwell paper. Strength of selection for males on the one axis, females the other axis. So here we consider a situation where we have two alleles, one that is full specialized towards the female optima, one that is fully specialized towards the male optima. Then we ask, can these two alleles coexist at a single locus? And then it's only when selection is symmetric or strong in the middle that they can coexist. So this model doesn't allow for evolution, it just checks whether these two can ecologically coexist. And here, if one add, this is under additivity. If we include sex-specific dominance, then the region of coexistence increases. So here is the result of my model. So the gray dashed line here, we still have strength of selection for females and males on the two axes, and this is the kind of reproducing the Kidwell result. But in this model, I allow for evolutions. With evolution, this intermediate allele can be introduced. It couldn't be that in the previous Kidwell model. And then we actually see that we don't get polymorphism in the end. So the color of this plot shows the number of coexisting alleles in the end. So it's only when it's strong and symmetric, and this parameter region actually mathematically correspond to the branching condition of Ewan's model presented previously. So this is under additivity. So we only get polymorphism when it's strong and symmetric. Otherwise, we get just one coexisting allele. However, when we include, so this is just a weak degree of dominance reversal, then we want, just as Kidwell predicted, we get this widened range of where we get allelic polymorphism. So we get polymorphism now also for weak selection. And there's something very interesting happening here when selection is weak and symmetric, in that we don't get two alleles, but we get many coexisting alleles. And here is the case when we have perfect kind of dominance reversal, maybe a little bit hypothetical, but then we get up to 17 coexisting alleles. So here we can see how it branches many, many times that coexist. And the reason is when you have dominance reversal, heterozygotes are the only ones that can benefit from these dominance reversals, because then they can choose the alleles that is better for their own sex. And while homozygotes have no benefit of a dominance reversal, this sex-specific dominance. So by having this very high number of coexisting alleles, then almost every individual ends up heterozygote and can benefit from this. And this will actually lower the sex load of the population a lot. Finally, we also allow for dominance itself to evolve. We model it as a separate modifier locus. And then we find that for most of parameter space, dominance do evolve, and when it evolves, we replicate the old result of getting this high number of coexisting alleles. So in conclusion, when we allow dominance to evolve, we see that when we allow sex-specific dominance to evolve, we see that we get this dominance reversal. And then when we have sex-specific dominance reversal, we get this large number of alleles, and it occurs under actually weak selection rather than strong selection, which was the previous requirement for getting high degrees of, getting allelic polymorphism under sexual antagonism. And this, the divergence that we see is here driven by heterozygote advantage, which is also an uncommon mechanism in these situations. You only get heterozygote advantage if you assume sex-specific dominance, otherwise heterozygotes are the maladapted ones, but here they are actually the adapted ones. Yes, so I want to thank my supervisors, funding, and you guys for listening.
Okay, we unfortunately, I think, are going to have to move on to Jason's talk. But if you'd like to ask Mattias questions, catch up with him at coffee, I'm sure he'd be happy to talk about this more. Let's thank him again.
Okay, hey everyone, my name is Jason Bertram. I'm at the University of Western Ontario here in Canada. And I'm going to talk about sort of an old school theory problem that I hope you'll agree is still interesting. And it may not be apparent at first, but I will get to genetic trade-offs. So I won't spend long on this. We've seen a lot of this today. So quantitative traits have lots of heritability. This is a classic study where they looked at lots of different traits, lots of different species. And Johnson and Barton summarized these numbers as saying somewhere between 0.1 and 0.6 is a typical heritability number. So that means a lower bound of about 10% of trait variance is genetic. So this implies a ton of genetic variation, right? And it's widespread. So this is a big question. Why is this happening, right? And by far the most studied hypothesis for this is the mutation selection balance hypothesis. And basically this just says selection's not doing anything fancy. It's removing variation. Drift is also removing variation. And mutation is replenishing variation. Hopefully that's enough to get us high heritability. Today this seems nice and plausible, right? Because we have GWAS studies and so on that tell us there's lots of polygenicity. And so it seems plausible, right? These quantitative traits are these huge mutational targets. So mutation is going to pump a lot of variation into them. So it sounds plausible. Let's run some numbers. So to do that, we need a model. This is the standard stabilizing selection model that all of you are familiar with. The main reason I'm showing this slide is just so you can see the symbols I'm using. So VS is the width of this fitness function. And then VG is genetic variance, V is the environmental variance. We don't actually have to pick a number for the environmental variance. We just sort of scale everything relative to that. Okay, and then for the other assumptions of my model, I'm going to follow what's known as the Ladder-Bolmer model. And this is basically keep everything simple, right? So we just look at one trait, we assume the optimum is standing still, everything is additive, there's no linkage to equilibrium, there's no genetic drift. Yeah, so this is kind of like vanilla mutation selection balance, right? We just keep everything as simple as possible. For those of you from a more quantitative genetics background, the predictions of this model are the same as the house of cards approximation. And as some people have mentioned today, in a model like this, new mutations are always deleterious, right? So stabilizing selection hates variation, new mutations introduce variation, new mutations bad. So these are the predictions of the Ladder-Bellman model. So I'm showing two curves here for different assumptions about the strength of selection. The upper curve is a number that was used a lot in the past. The lower curve is a number that Johnson and Barton said we should be using, and we've been cheating by using the upper curve. And I'm plotting the heritability versus the target size. So in other words, the number of loci that contribute to the trait. But instead of just showing like numbers of loci, I've scaled it relative to the total euchromatic genome, in this case of Drosophila. And I'm assuming a Drosophila mutation rate. So what you can see is that to even get to the lower range, so that the shaded region is the range 0.1 to 0.6 for heritability, to even get to the lower bound of this region, we need something like 1% of the entire euchromatic genome for one trait, right? And we don't just need that for one trait, we need that to be a typical value for like traits that we see like widespread in nature, right? So this is starting to seem a lot less plausible, right? There's a deeper issue here as well, which is that maybe we choose a different species and we have some slightly different numbers and we can shift the curves a little bit and we can convince ourselves. But the deeper problem is that the model is just too sensitive to parameters that we know will differ, right? And we're trying to explain a pattern that's like fairly consistent. And so there's a qualitative problem with the model, right? It's not giving us consistent numbers, it's got this huge spread in. And so it's easy to find traits or species where the numbers are lower and then it breaks, right? This is just the Ladder-Bellman model. There's lots of other mutation selection balance models that are more complicated. They all have similar issues. So today I'm going to take this classical picture and I'm going to change one thing about it. And that is that I'm going to say that the optimum is not allowed to be still anymore. So this is like obviously true, right? We know that trait optima do move around. The environment is not stationary. And this actually changes the model in a fundamental way. So instead of having new mutations always be deleterious, your fitness effect now depends on the environment, right? So the trade-off part of this is like almost a logical trade-off. Like a mutation cannot both increase and decrease the trait value. Therefore, it has to pick to help or hinder if you're trying to get to an optimum that's somewhere, right? The upshot of this is that mutations can now be beneficial. They're not always deleterious. So I'll get into the consequences of that in a moment. First, let's just talk a little bit about the movement of the optimum a bit more. So, like, it's obvious, right? Nobody's surprised to say that the optimum can move. The hard part comes when you have to specify how the optimum moves, right? And that's where previous models have really struggled. And so in particular, probably the most important work that's been done on this in the past has focused on a cyclical optimum change. So maybe you're thinking of something like a seasonal cycle. And the predictions of these models are notoriously sensitive to the parameters of your cycle, right? So if you have big amplitude with long period, you get totally different predictions to short amplitude and so on. And so that it's just really hard to see how this will help us to explain anything, right? So what I'm going to do today is I'm going to kind of cheat. I'm not going to come up with some brilliant new model. I'm just going to copy a model that is already used to describe how trait optima move. And that is a Brownian motion walk, or different kinds of random walks really can be used. So these are routinely used in phylogenetic comparative methods to describe how trait optima move over time, right? And so we may not fully believe that they're 100% accurate, but at least they're like used in practice on data, and we can, there's a direct connection to the empirical world there. So if I just assume a simple Brownian motion random walk, I've only got one new parameter in my model, and that's the rate at which variance accumulates per generation, which I'll use a sigma squared for. So basically that number is a measure of how rapidly the environment is changing or the intensity of environmental fluctuations. Yeah. Okay, and then just a reminder, I'm going to keep everything else the same. So same classic ladder-Bolmer model. I've crossed out that the population, the optimum is constant, except for one thing that I just couldn't let go of, which is not having drift, because that's just silly. So I put drift back in. This doesn't help me at all. Drift is strictly worse for variation. So this actually makes my life harder, but I wasn't comfortable leaving that assumption. So other than that, let's keep everything as simple as possible. Okay, so these are just some simulations to sort of illustrate what happens. So this is a very intuitive, straightforward thing. On the left, when the optimum is stationary, the mutations are always deleterious, they're kept at low frequency. On the right, they're sometimes beneficial, so they can rise to higher frequencies. More mutations at higher frequencies equals higher genetic variation. Great. The actual predictions, if you compare them to the ladder-Bolmer model, look a lot better, right? So the dashed curves here are the case where the optimum fluctuates. Everything else is the same. These curves are actually calculated with a lot of math that I will not talk about at all. But so you can see that it's improved the predictions a lot. So we're basically sticking in this region we want to stick in for a much broader range of parameters, especially on this lower end, where we can believe that target sizes like that are more typical, right? The other thing to notice is that there's also much less of this sensitivity issue, right? So the curves have been flattened out a lot at the lower end, where we have the ladder-Bolmer model really dropping off, right? And then one more comment I'll make on this is, to get these results, I didn't assume a rapidly changing environment, right? So I chose a very modest rate of change, so that 10 to the minus 2 there means that the rate of accumulation of trait optimum variance is 1% of the environmental trait variance per generation, right? So the environment's not racing away here. And remember, it's also not moving consistently in one direction. It's kind of wandering around, and every now and then it gets away from you, and you have to catch up. Okay, so this is all well and good. I mean, obviously, it's a really simple model, right? So who cares, right? The interesting part is not that you can get the numbers to work. The interesting part is, why is it so much better? And maybe that has some insights about what's going on in nature or what would happen in more complicated models, right? And so the key insight here is that we've spoken about how fluctuations can create genetic variation, but there's a reverse of this, which is that the fluctuations themselves are determined by genetic variation. And to see this, you just got to remind yourself of Fisher's theorem, the Breeder's equation, et cetera, right? So the rate at which the population chases the optimum is proportional to the genetic variation that it has available to chase the optimum, right? And so the relationship between the strength of fluctuations and VG is not simple at all. It's this complicated sort of circular thing, right? And so, you know, if you imagine a scenario where a population has low genetic variation, it can't keep up with the optimum, so the optimum can really run away from it, and it can get really strong selection coefficients on alleles that help it to get back to the optimum, right? So it's going to experience very intense fluctuations and potentially a very large boost to its genetic variation due to fluctuations. And vice versa, if a population has high genetic variation to begin with, it's almost like a stationary optimum model again, like it just tracks the environment so closely that it doesn't matter at all. So in sort of the terminology of like feedbacks, this is what we call a stable feedback loop. So you've got environmental optimum fluctuations cause selection coefficient fluctuations. Selection coefficient fluctuations will tend to increase genetic variation, but genetic variation tends to dampen selection coefficient fluctuations. And so this narrows the range of genetic variation predictions that the model will spit out, because there's sort of a happy range that it wants to be in. None of this is possible without putting in just that key thing of not allowing the optimum to move. So I'll get to my take-homes. So yeah, so the main thing here, I guess, is like people have been hating on those mutation selection balance models for a long time, including Nick Barton himself, who did a lot of that really good work in it. And you can actually, there's a thing you can change that's very realistic that actually helps a lot. The key thing that we needed to make this work is that the trait optimum needs to move in a specific way, namely more like what we see in a phylogenetic comparative method model. So it's sort of wandering through space, and the scale of that wandering needs to be like a decent size compared to the variance in the trait itself that you'd see segregating in the population at any given time. But the key thing is that doesn't have to happen very quickly, right? So it can wander away pretty slowly, as long as it's not like too slow, which that's a precise statement that I'd have to justify in much more detail. And then the mechanism that causes this to happen is quite generic, right? It's not tied to any of the simplifications I made in the ladder Balmer model. Presumably something like this will apply in more complicated models, and then you could hope that perhaps this is something that's going on in nature as well. Okay, yeah, I'll leave it there. Thank you.
Thanks, Jason. I think we have time for maybe one or two questions.
Yep.
So in your moving optimum model, would you get different results? So your mutations affect a single trait, but in reality they affect lots of traits which are under, potentially, because you were like, the mutations can become good because they move the optimum, but they would potentially move other traits and still be mostly bad.
Yeah, so then essentially it's a multidimensional thing, so you have some vector, and so there'd be a similar higher dimensional version of that, right? So the good ones move you closer, the bad ones move you further away.
And your trait could just be multivariate trait optimum
Yeah.
or something.
And you really have that view in mind when you're thinking of a Brownian motion type thing, right? Because it's not a simple thing of, okay, hot, cold. It's like, okay, now there's like a slightly different virus in my environment, and also my population density went down, and like a new species of grass is around me, and so you're imagining sort of a higher dimensional type. Yeah. But yeah, obviously, like you would ideally work in higher dimensions for a more complicated. Yeah.
So thank you so much to Luke and Carl for organizing the symposium and inviting me to speak during it. I'm really excited to talk to you today about one of my favorite things, which is the fern gametophyte, and how we're using it to look for evidence of gene conflict between life stages in ferns. One moment while I figure out how to do this. Okay, so a quick overview of the talk. I'll begin with an introduction to land plant life cycles and life stage dominance for a little context, and then I'll briefly show you some results from simulations of gene conflict that I generated using a program that I wrote called Shady. And finally, I'll show you the data from the developmental transcriptome time series, and I'll show you some preliminary analyses that we're working on looking for evidence of conflict in this system. Okay, still figuring this out. All right, so I'm assuming most of you are coming from an animal background, and if you're not, you are an animal. So as you know, most animals have this dominant multicellular diploid life stage, and so they spend the majority of their lives as a diploid and a relatively short part of their lives as a haploid cell in the form of the sperm and the egg. And you'll sometimes see this referred to as a diplontic life cycle, which refers to the dominance of the diploid stage. But plants do things a little differently. So they also have a multicellular diploid stage, just like animals. But instead of making gametes, the sporophyte body makes spores. Those spores are haploid, and they germinate and grow into a haploid body, and this is the gametophyte. And so this is a separate haploid multicellular structure, and it's actually the gametophyte that is going to make the gametes, and then the sperm will fertilize the egg, and the life cycle proceeds as is familiar to us with a diploid zygote. And so the plant life cycle is often referred to as alternation of generations, which is shorthand for alternation between two multicellular life stages with different ploidy levels. And for those of you that have never seen a gametophyte before, here are some photos of fern gametophytes. In all plants, the gametophyte is the haploid part of the life stage, and it creates the gametes. But in ferns, as you can see here, the gametophytes are also photosynthetic, and though they might look quite delicate, they can actually persist in the environment for weeks to years, and they live independently. And so, also, though they're not as showy as the sporophyte, they also have a diversity of forms that they occur in. And so the feature of the fern gametophyte, that it's completely independent from the sporophyte, is actually a very unique condition in land plants. Here we have all the major lineages of land plants with a little cartoon of their life cycles. The diploid part is in green on the top, and the haploid part is in red on the bottom. So in bryophytes, there is an independent gametophyte, but their diploid sporophyte is not independent. The sporophyte grows on the gametophyte, relies on it for support and nutrition, and eventually dies, and the gametophyte can live on. And so the haploid gametophyte is considered the dominant life stage in these groups. In flowering plants, we have exactly the opposite. So it's the diploid sporophyte, that's the dominant life stage, and the gametophyte is highly reduced and completely dependent on the sporophyte. And interestingly, this looks a lot like convergence on an animal life cycle. It's almost diplomatic, but that's a subject for its own talk. So the independence of both life stages in ferns allows us to study them separately, their gene expression, morphology, etc. And this is something that we just can't do with other lineages. Okay, so this part I added after I was accepted to the symposium, so I apologize, I'm gonna really blaze through it, but I thought that this audience might find it interesting. So if we think about the fern life cycle from a theoretical perspective, it raises some really interesting implications for how new mutations are going to fix in this system. Because during the diploid part of the life cycle, new mutations are heterozygous, and so they're going to experience dominance and masking effects. But during the haploid part of the life cycle, there is no heterozygosity, and so all of the new mutations are going to be exposed to selection right away. And so a few years ago, I wrote a Python program called SHADY, which stands for Simulating Haploid-Diploid Evolution. It's a wrapper around the very powerful forward simulator SLIM and the coalescent simulator MS Prime, and it uses a hybrid forward-backward time approach to efficiently simulate complex life cycles, like the life cycles of plants, and this just makes it feasible to explore theoretical questions in these systems, whereas otherwise it would be very computationally intensive to simulate. So mostly as an exploratory exercise, I wanted to look at what happens when gene conflict occurs between different life stages. So traditionally, gene evolution is modeled with this simple assumption that one gene affects one part of the life cycle, but what happens when a gene affects both parts of the life cycle, and especially what happens when the selective pressures are opposite in those two life stages. And so this is what I mean when I use the term gene conflict in this talk. So to give you an intuitive sense for what we learned from these simulations, I'm going to start with results from a typical Wright-Fisher model and build in modifications. So these are simple simulations where I just introduce a new mutation with some selection coefficient from negative 0.3 to positive 0.3, and then I measure the probability of fixation of that mutation over many replicates. So in a Wright-Fisher process, the probability of fixation is proportional to the selection coefficient, and therefore mutations with negative selection coefficients, they generally don't fix. And another feature of this model is that the population size doesn't affect the probability of fixation. So in these graphs, each of the different colors are different population sizes, but here you can see the probability of fixation is essentially the same between them. However, when we add in alternation of generations, we see that the probability of fixation of new alleles is now impacted by changes in population size. So in these simulations, we're holding the size of the diploid population constant at 1,000 and varying the gametophyte population size, and all of the new mutations are neutral in the sporophyte life stage, and the selection coefficient is only affecting the haploid life stage. So as you can see from these results, changing haploid population size is now causing differences in fixation probability of new alleles, but what we're really interested in is gene conflict, so what happens when we add in conflict between the life stages. Okay, so here the selection coefficient of the new allele is set to be highly beneficial in the diploid sporophyte, it's 0.5. And the population size is still being held at 1,000 diploid individuals. And again, we're varying the selection coefficient in the haploid gametophyte from negative 0.3 to 0.3. And what we see is that mutations that have a negative effect are now able to fix. So ultimately, this suggests that, at least from a theoretical perspective, if gene conflict is happening, it does affect genome evolution, and it results in different outcomes for new alleles. Specifically, it causes mutations with deleterious effects in one life stage to fix, whereas they would have been weeded out by selection otherwise. And so the question that we have now is gene conflict actually happening? Does any of that really matter? And so that brings me to our empirical data set, where we're using this transcriptome time series to look for evidence of conflict. So very briefly, I collected 6 time points during gametophyte development in the model for Enceratopterus retardii. In the first 3, the gametophyte is the only tissue present, and then by the 4th time point, the fertilization has occurred, and so the diploid embryo is also present in those time points. And then I also have data from fertile and sterile leaf tissue from an older sporophyte. And so we sequenced these time points using bulk RNA-seq. For a time, I'm not going to talk about the bioinformatics pipeline. So if we just look at presence-absence of genes to start out with, I have 4 groups here. There's 2 gametophyte only in red and yellow, the mixed tissue in purple, and the diploid sporophyte in green. And really, the whole point that I want to draw your attention to is how much these circles are overlapping. So there's about 26,000 genes in total in this data set, and you can see the majority of them are expressed in both of the life stages, so over 80% of the genes are present. Of course, this Venn diagram is only showing presence-absence in the different tissues, but genes can also be present in multiple tissues, but expressed at very different levels. And so this heat map is showing a subset of the genes that were most differentially expressed between the sporophyte-only time points and the first gametophyte-only time point. And it's really just a nice way of visualizing how different the transcriptome profiles are between the tissues, even when the genes are technically being shared between them. And it's actually this differential expression that we're going to try to leverage to look for evidence of conflict. So we can take our differential expression analysis, and the volcano plot is just a nice way of understanding quickly how we're using it. So all of the genes that are upregulated in the diploid are on the left, and all of the genes that are upregulated in the haploid are on the right. And then a log base 2 fold change of 1, which is where the lines are drawn, corresponds to a doubling of expression in one group compared to the other. And so everything that's shown in red is passing that log 2 fold change threshold, and also a p-value threshold. And typically these would be the genes that people are interested in when they do a differential expression analysis, because they are the genes that are differentially expressed. But for our purposes, we are also going to make use of this group in the middle, which are the genes that are passing the p-value threshold but are not considered differentially expressed between the 2 groups. And so this analysis that we're doing is based on the hypothesis that if genes in this fern are experiencing conflict between life stages, then we expect to see a correlation between gene family size and gene expression patterns. The logic behind this assumption is that the bigger the gene family, the more that family has undergone duplication in its history, which means that there was more opportunity for it to undergo subfunctionalization. And if there's gene conflict, then there should be some selection pressure for subfunctionalization to lead different copies of a gene to be primarily expressed in one life stage or the other, thus reducing conflict between life stages, because as we saw, the presence of conflict can cause deleterious effects in the genome. And so we use the differential expression to determine which genes are mostly expressed in one life stage or the other, and which ones are considered shared. And I chose pretty strict bins for this, so the sporophyte dominant genes are at least 100 times more expressed than the gametophyte, and vice versa for the gametophyte dominant genes. And then for a gene to be considered shared, the fold change needed to be less than 40% different between the 2 groups. Okay, and then determining the gene family size has been more tricky for this organism. This is technically the model fern, but the genome was published less than 2 years ago, and so we don't really have the wealth of resources that most model organism researchers enjoy. So before we invested a lot of effort in solving the problem of robustly determining gene family sizes for Ceratopterus, we just wanted to see if this idea held any water. So I ended up using PFAM as a very rough approximation this information was already available as part of the genome annotation. And I just ended up counting the number of genes in the entire genome with the same PFAM set. So for each unique set of PFAMs, we just have a count of how many genes were annotated by that same set. We're pairing that with our gene expression bins, and this slide is just to warn you that I couldn't help myself, and I added 2 more bins of like semi-sporophyte dominant from -100 to -10, and semi-gametophyte dominant from 10 to 100. So you'll see 5 different bins in the data, and here they are. So the 5 different bins based on the fold change with family size on the x-axis, and the histogram is showing the frequency of each of these family sizes. And I think this evidence is actually really compelling. The pattern is stronger than I expected it to be. So we're seeing a much higher frequency of smaller families in the shared gene bin compared to the life stage specific bins. And then this effect seems to be more pronounced in the gametophyte life stage, which may be related to its haploidy. And so to me, this suggests that there may actually indeed be conflict between the life stages in ferns. And so we're planning on doing a lot more work to perform this analysis more robustly. And we're also interested in bringing in evolutionary context to look at when the genes duplicated and whether there's some signal based on whether duplications occurred before major life cycle transitions. And we also have a comparative data set that we'll use to look for signatures of haploid versus diploid selection and whether that correlates with these expression bins as well. So that's all I have for you. Thank you so much for attending my talk.
Wonderful. Thanks very much, Elissa. I think in the interest of time, we should get set up for the next talk, but I'm sure if you find Elissa, she'd be happy to take your questions, and thank you very much again for an interesting talk.
First of all, I also want to thank Carl and Luke for organizing this super cool symposium. I learned a lot already today. Second, I want to give a warning. Unfortunately, I have just one genomics slide in my presentation, and I will make sure that you don't miss it. I will mention when it is there, so it has a few data. Thirdly, I'm happy to present a few very hard-earned fitness data from a wide vertebrate population, so I hope this will be of interest for you. And among them, there will be also a very illustrative example, I hope, of extremely strong sexual antagonism, which is perhaps new for some of you, this example at least. And lastly, if you're still not convinced, stay for the pretty pictures. So, for the introduction, you heard this already quite a lot today. So there's the genome, and when you have diversity in the population, for example, introduced by the sexes, then there can be different reproductive roles, and this can cause conflicts at the genomic level, which has to be resolved. And the conflict is caused by the different evolutionary interests of males and females, which lead to different fitness optima. And the resolution of this conflict can come in intralocus, at the locus level, for example, by specific alleles that are either benefiting the males or the females, or if it's going further, even at some stage, there can be more differentiation and entirely different sex chromosomes that help to mitigate this conflict. It's not only that there are discrete classes in populations like sexes, some species also have some more discrete reproductive morphs, and here it's the same analogy. It's just in terms of reproductive roles, think about mating strategies, and again, there will be loci which might encode different alleles that either benefit one or the other morph in this case. The question then, of course, is what are these loci? And in the past decades, chromosomal inversions or so-called supergenes have been very, very prominent in perhaps representing these loci. And that is because they're very often associated with reproductive diversity, with discrete reproductive variation in species. A couple of examples are here. And what is also thought is that balancing selection is maintaining these inversion polymorphisms, but the exact mechanisms of balancing selection, so what it is, heterozygous advantage, negative frequency dependent selection, sexual antagonism, and so on, it's often still unclear. So we work with a very illustrative species, the ruffs. As you can see, it's a bird. And these ruffs come in 3 different morphs. And there's a little short video starting in a moment, which will introduce these morphs to you. First of all, you see here these some males of the independents. These are large males. This is also the most frequent morph, about 80 to 90% of them. And the second morph looks very similar, but they have light plumage. These are called satellites, about 10 to 20%. And the third morph is looking like females. These are faders. So independents are aggressive. They defend a territory in a mating arena, and they display together or sometimes also with satellites to try to attract females. And these females then flock into the mating arena, and then they go either to a territory of an independent, or if there's a satellite and the co-display happening, as you can see here, that is super attractive, attracts many females that all come and queue on this territory for copulations. And of course, there's lots of conflicts on there, so that's how the satellites also get their copulations. But also the very rare sneaker morph is really good in using these conflict situations, and then comes occasionally to copulation here. And these sneaker morphs, these faders, are extremely rare. It's about 1% only in the population, so this camera team got very lucky when they filmed that. So just to summarize once again, we have these three morphs, and there's this color scheme here which I used through the presentation. In blue, the independents, the aggressive big morph. In red or purple, the satellites, the cooperative morph, which is also displaying. And in yellow is the faders. These are the sneakers, which are not aggressive and also not displaying. And here's a genomic slide. So underlying these three morphs, three mating morphs, is this supergene, a chromosomal inversion. It's autosomal, it's dominant. And what is really important, it only can be existing in a heterozygous form, because one of the breakpoints of the inversion runs through a gene which is important for mitosis. So it's the breakpoint is homozygous lethal. So these combinations here of double satellite alleles or double fader alleles and so on, or a mixture of them, that don't exist in nature. So I mentioned that the supergene is really encoding these different traits for the males. However, it's autosomal, so it appears also already in chicks and also in females. And the question then, of course, is, does it have fitness consequences or any consequences for these life forms too? Yeah? And that's what we looked at over the last few years. So the first project was done by graduate student Lina Giraldo-Deck, who finished her thesis already 2 years ago. And she asked whether these different superalleles somehow have effects on the reproductive success of different females. And she used for that a captive population in Canada, because in this population we have lots of these different morphs. So also the rare morphs are kind of in an acceptable proportion. And what she found is indeed that they have reproductive consequences. So especially for the faders, the very rare morph, they lay fewer eggs, as you can see here, in different years. Across different years, it's consistent. And there's also senescence effect, so that's why it's a quadratic. And they also lay smaller eggs than the other morphs. And what she found then when she looked at the survival of the embryos and of the eggs is that the maternal morph actually was the one which was determining the survival. So the fader females, again, had very poor reproductive success. Their eggs had a high mortality. And going on then from once the chicks had hatched, it was not the morph anymore, but rather the egg size that determined survival until fledging. And here again, the small eggs are doing much poorer than the large eggs. And as I said before, the faders are producing smaller eggs, so the faders had a disadvantage here. And when you sum this all up, which is shown here in this graph, and standardize it to an ordinary independent female here in blue, 200 eggs, you see that the faders are losing out the females at every every stage, until hatching, until fledging, and also until fledging. So in the end, they only have about 1/8 of the reproductive success of an independent female. And so then, of course, there's the question how this can be maintained in a population if it's so bad for these females. And the solution is that the males are actually compensating for that, and they compensate by a higher than average mating success. They have we modeled that it's about 3 times higher mating success than the independent males. Yeah. So why the fader gene doesn't disappear, that's here a little model for that. What we did is it's a combination of sexual antagonistic selection and also negative frequency dependent selection. Why negative frequency dependent selection? It all starts with a mating advantage for fader males when they are rare, because they're a snaker type and the other roughs can identify faders otherwise and throw them out of the leg. And when they have this mating advantage, they produce more fader offspring, which is then bad for them, so the advantage decreases. And at the same time, it's also that the fader females reproduce poorly. So that again leads to the reduction in the allele frequency, so there's fewer fader offspring, and the whole thing starts again in there. So much for the females. So the next question is, then, does the super allele also affect the survival and also the maturation of the chicks? And this is a combined work of Lina Giraldo-Deck again, and also of James Tolliver, another PhD student in our lab. And for that, we wanted to get fitness data or survival data from the wild. And that's a picture what you have here. So on this picture, you see 4 data points. I remind you that we're working with birds here, not with some plants. You're looking at the rough nests, yeah? Of course, the ornithologists in the audience already spotted it. For the others, I have magnified it once again. It's in there. And we did this a lot of years. It was a lot of effort to find these rough nests on these Finnish meadows, and we ended up with in total quite impressive sample size. I think from 2016 to 22, we sampled over 780 embryos or chicks from more than 200 nests. We also radio tagged a few of them for survival, but the radio tags were a bit heavy. So, and from that, at least, we got the frequencies at the initial frequencies of the morphs and the sexes, and these are listed here. So, as I said before, the independents are the most frequent morphs, followed by the faders and by the satellites and the faders. And the faders we didn't have in the first 5 years, so we got lucky that in the 6th year when we found finally some fader offspring in these nests. Anyway. So we couldn't really follow the morphs and their survival and get specific survival data in the wild. What we did then instead, we hand-raised a lot of rough chicks for that and got the survival data from there. And more importantly also, we got also fletching data, because fletching is really an important milestone in the species, because then they can escape the ground predators. And here you can see the setup of the fletching test. It's very ingenious. So there's a pot here. And then the chick has to jump into the water or fly over this water basin. And if it can fly, then if it can jump over it or fly over it, then it can fletch. And we did this every day when they had the wings developed. And here is a distribution of the fletching ages, the sexes here in triangles. And what we did see there is that the males fletch later than the females. And there was also, in males at least, the morph separation. The independents were the last and the faders were the earliest. So not so much differences in the females. And then James went to work and used all these data, what we had on survival and on fletching, and constructed a big population matrix model using our data from captivity, some are from the wild, and also got some literature data on a fletching effect, how much would that boost survival, and so on. And here are briefly the results. When you look at the morphs separated by sexes differently, you don't see much. So you don't see any changes in morph frequencies from hatching to fletching. Oops. However, when you look at the males, at least, there's a small change. Independence decrease slightly, and satellite males increase, but not overwhelmingly strong. However, there's really strong effects among the sexes. So the males are actually doing worse with time. So they don't survive as well, and they also fletch later. And in the end, the male ratio decreases from 50% in the population at day 33 at fletching to 40%. So it's quite something, and it's about 2 females on 1 male. And with that, I'm already at the end. So briefly to summarize what I showed you today. So this polymorphism at the supergene is maintained by balancing selection, and it's an interplay between negative frequency dependent selection, sexual antagonistic effects for female faders at least. The super alleles have really strong sex-specific effects that modify this intralocus conflict, and these antagonisms can also change between life stages and are not the same in there. And finally, that these antagonistic effects have some early life effects already that change population demography, especially the sex ratio in this population. And also the males are more vulnerable than the females before maturation. And with that, I'm ending here, and I want to acknowledge and thank all the co-authors and the many people who helped with that. And if you have any questions, then go ahead.
Couple of minutes for questions.
Say it again.
What does this locus actually call?
So there's about 100 genes in there. Among them, some genes that regulate steroid hormone levels, for example, in the blood. There was just a talk today, a fantastic talk of Alex Sabella here, a graduate student, how this works. And also a few other genes like transcription factors and one gene which is associated with telomerase regulation and things like that. So a wide subset of things.
So I guess because of the conversion, so it's sometimes this recombination.
Absolutely. In this case, recombination is extremely rare because it actually only exists in heterozygotes, right? That's what I said. So it's hardly ever happened. One rare event actually created the satellite morph in a recombination event, but other than that, there's nearly no recombination going on. So these are really going in different direction, which is also consistent with loss of function. When you look at the fader phenotype, for example, it lost the ornaments, it lost the courtship display, and also the aggression. Yeah, Nathan?
So, I was curious, is anything on the inversion related to the normal sort of size dimorphism that you see within Rus, but is different than all the other shorebirds where you have reverse sexual size dimorphism?
No, actually the sexual size dimorphism stays within each morph the same. It's always 70%, yeah, in that case. So there's nothing in there which modifies it. Yeah.
You mentioned that there's like an age effect.
Yeah.
Females decrease in the number of eggs they lay and the clutch size as they age.
So the clutch size stays the same, but they would lay fewer eggs. We always remove them when they laid the eggs in the aviary, and then they would, after 6-7 years, so they're quite long-lived, can live up to 16-20 years. After 6-7 years, you have the peak, and then it goes down afterwards.
And do you have any sense that the aging trajectories are changing between them all?
In that model, we couldn't do it. We couldn't allow for different things and that. It's possible that it does. We do have some data on survival in adults that suggests that. It's not straightforward, something like that. So I believe the satellite has actually have the highest survival from that in adults.
I will have to change to the next speaker now. Thank you.
Okay, hello everyone. My name is Roman. I'm a postdoc in Hanna Kokos lab at the University of Mainz in Germany. And I will be talking about the Mathieu effect in biology, or do social insect queens really reverse the fecundity longevity trade-off? So the trade-off between survival and reproduction is one of the most fundamental trade-offs in life history theory, where a higher investment into reproduction results in a shorter lifespan. So if you plot lifespan against fecundity, this results in a negative correlation between both traits. And of course, the reason for this trade-off is that there's only a limited amount of resources that need to be allocated into conflicting functions, in this example, between reproduction and survival. But here come social insect queens that seem to escape this trade-off. Because it is well known that queens lay thousands or even millions of eggs and live for years or even decades, whereas workers are usually non-reproductive and have much shorter lifespans. So if you plot the same relationship between, if you plot the correlation between fecundity and lifespan for social insects, it turns out to be a positive one. And this makes for an interesting contrast. Whereas life history theory predicts a negative correlation between fecundity and lifespan, meaning that the trade-off exists and is clearly visible, social insects show a positive correlation. So one could say that here for social insects, this trade-off is masked. And in social insects, the masking of the trade-off actually occurs in two different contexts. The first context we have just seen, that is the one between between queen and workers. But this one is also not really surprising if you think of the colony as a superorganism, right? With distinction between germline and soma. So the other context is actually more challenging to explain, and this is the context, the comparison between different queens, where those queens that are observed to be very productive are also the most long-lived ones. And the question, therefore, is how to explain the masking of the trade-off in this second context, when you compare different queens in different colonies. And to tackle this question, it's important to remember that it's not all about resource allocation, but also about resource acquisition. So if you imagine you are able to acquire more resources, this means that you also have more resources to allocate, both into reproduction and survival. In other words, high levels of resource acquisition can mask this trade-off. And this is by no means a novel finding. It was nicely explained in the seminal paper by Van Noordwijk and de Jong almost 40 years ago. Now, how do differences in resource acquisition originate? Well, it is well known that these differences can arise through variation in individual traits or quality. And here in this example, I just chose habitat quality, so where the left colony happens to be located in a good habitat and the other colony in a not so good one. But there's also another explanation, and that's where the focus of my talk is, and that is basically just luck or chance. And it will become clear what exactly I mean by luck in this context. And actually, differences in resource acquisition that are due to luck are already known in the Bible, where it says, "For to everyone who has will more be given, and he will have abundance. But from him who has not, even what he has will be taken away." And the rich get richer dynamics, or this self-reinforcing accumulation of advantage, is well known as the so-called Mathieu effect. And I will argue that this Mathieu effect also exists in social insects. So if you consider a lucky queen that happens to have more workers, then this lucky queen is also able to acquire more resources, which means that she has also more resources to allocate into longevity and fecundity, leading to even more workers who then can acquire even more resources, and so there's a positive feedback loop, which pretty much resembles the Mathieu effect, the self-reinforcing dynamics of the Mathieu effect. Whereas if you have a not so lucky queen with fewer workers, there will actually be no positive feedback loop. Or if there is one, it will be much slower. Now, we propose a simple model to tackle the following questions. First, how easily does the Mathieu effect mask the fecundity-longevity trade-off in social insects, which would be visible as a positive correlation? And second, how easily can this masked trade-off be unmasked, which then would be visible again by a negative correlation between fecundity and lifespan? And I will not bother you with the details of the model. I will just mention the most important model assumptions. And that is first that worker and queen lifespans are stochastic. That's basically where the luck or the chance component comes into play. Further, we assume that queens with more workers have a larger resource budget than queens with fewer workers. And then a queen basically makes two fundamental life history decisions. The first one we denote by X, which is the proportion of resources that are allocated into reproduction, and therefore a proportion 1 minus X is allocated into the queen soma, or her maintenance, her longevity. And the second fundamental decision that a queen has to make, we denote by Y, which is the sexual threshold. What is this? So the sexual threshold is the number of workers above which sexuals are produced by the queen. So we assume that queen starts to produce only workers, and once the number of workers has reached a certain size, which is given by Y, then the queen switches to producing sexual eggs, okay? And therefore we define queen fitness by the lifetime production of sexual eggs. Now, we first asked what values of X and Y maximize queen fitness. And so what you see here, please have a look at the right panel. So what we plotted here is queen fitness, that is the lifetime production of sexual eggs, as a function of allocation to reproduction on the x-axis and sexual threshold y on the y-axis. And the result is not really surprising. It turns out that queen fitness is maximized when allocation to reproduction is relatively high and when the sexual threshold is relatively low, which means that the queen starts relatively early to produce sexual eggs. And the left panel is just to show you that because the model involves stochasticity, so the left panel shows the result of a single simulation run, whereas the right one shows the results of 100 simulation runs. And you can see that if you only have one simulation run due to stochasticity, it is possible that even if the queen chooses a relatively optimal combination of x and y values, she might end up with low fitness or even zero fitness if, for example, she dies relatively early, right? That you can see by the blue spots in the otherwise yellowish area. But now to the crucial question: how frequently are positive fecundity longevity correlations observed? And the answer to this question actually depends on, or in our model at least, depends on two choices. The first one concerns the question of what values of x and y do queens use. And here we make 4 distinctions. The queen may use optimal x and y values. The queen may use only optimal x values, only optimal y values, or suboptimal x and y values. And the reason for this distinction is that we expect that optimal x and y values should mask the trade-off, whereas perhaps with suboptimal values we might have a chance of unmasking the trade-off, right? And then there's another question, and that is what measure of queen fecundity do researchers use? And here again we make 4 distinctions, and these are total x over the queen's lifetime, sexual x over the queen's lifetime, or total x as a rate, sexual x as a rate. And that's basically because researchers actually do use different measures. So by these distinctions, we try to facilitate the comparison between our model results and what researchers actually look at. Okay, so what you see in this figure is you see 16 panels, 4 rows, 4 columns, and each row, let's start with the columns. Each column shows one of the 4 distinctions of what values of x and y a queen may use, going from optimal values on the left to suboptimal values on the right. And then each row shows the 4 distinctions we made regarding the measure of queen fecundity, right? Total x over the queen lifetime, sexual x, and then total x as a rate, sexual x as a rate. And so in each of the 16 subplots shows basically the correlation between queen lifespan on the x-axis and queen fecundity by different measures on the y-axis. And the main takeaway from this plot is that actually positive correlations between fecundity and longevity are very common. However, so far we have only looked at one particular set of parameter values. So if you want, you could think of this one particular set of parameter values as depicting one hypothetical ant species. Okay, and therefore the question, of course, arises, what happens if we change these parameters? How do correlations look like for different parameter settings? And what we did here is basically you see the same 4x4 figure, but now we plot in each subplot the correlation between fecundity and longevity. Blue means the correlation is positive, orange means the correlation is negative. And we do this for 50 randomly chosen parameter settings, sort of 50 hypothetical ant species. And here the main takeaway is that you mostly see positive correlations, and you only see negative correlations if queens use both suboptimal X and Y values on the rightmost column. So the takeaways are differences in resource acquisition can mask life history trade-offs, as shown by this paper, Van Noordwijk and de Jong. The Mathieu effect easily masks the fecundity longevity trade-off in social insects, and unmasking this trade-off might actually be quite difficult by observational data alone. And by this, I'm done. Thank you very much for your attention, and I'm happy to take any questions if there's time.
Thank you very much.
Oh, hello everybody. So my name is Mathieu. I'm a postdoc at the University of Toronto, and today we're going to talk about quantitative genetic. And I do this work in collaboration with Professor Jacqueline Stepanak at UFT. And so something I like to study, it's genetic correlation in general, and today we're going to talk about the effect of asymmetry of cross-sex genetic correlation in the evolution of sexual dimorphism. So for the people who don't know me, I really like to study sexual dimorphism, and I find that very interesting how much male and female can be different, and how much this different can be very variable and very diverse across different species, going to insects to mammal species. And so in general, what we can see, difference in male and female are more in morphology, but we can find this difference also in physiology and behavior, and also in life history. As we know, female have a longer lifespan compared to male. So in general, what we can say in nature we have a large variation and diversity of sexual dimorphism across species. And the way I see it, it's more simple. I see it as a bimorphic distribution, when the red population in the female one have a higher mean compared to the blue one. And something, from my opinion, which is very interesting, it's how in evolutionary biology, it's how we go to this monomorphic distribution to a bimorphic distribution, knowing the fact that male and female have a lot of genome in common. So to answer this question, as everybody has known now, we have this very famous quantitative genetic framework when the presence of antagonist selection, so distinct direction between male and female selection gradients. So in this panel, we can see the female population have a kind of a positive selection gradient, whereas the male is more a negative one, so we have opposite direction, and that will produce sexual dimorphism at the end when each population can reach their fitness optimum. But this phenotypic dissociation needs to be also associated to a genetic dissociation, and we can quantify that in a quantitative genetic framework by estimating the cross-sex genetic correlation, which is called RFM. And to have this dissociation at the genetic level, so we can see the RFM quantifies the share amount genetic variance between male and female, and by decreasing this correlation genetic, we can see a dissociation at the genetic level between the sex, which can lead to this sexual dimorphism per se. And for the people who study that, in general, what you can see in the center, we have antagonist selection and also a strong genetic correlation between male and female, and that can show how much shared genetic variance can constrain the sexual dimorphism. And something we can challenge a bit of this framework, but not thinking about antagonist selection per se, but concordant selection, which male and female have difference not in direction, but in magnitude. So here in this example, we can say the female one have a very strong magnitude of selection, and that will produce sexual dimorphism at the end also. And why I'm talking about that, it's because when we look at the different meta-analyses on selection gradient in the literature, what we can see in general, antagonist selection is pretty rare. And also asymmetry, so difference in magnitude between male and female in concordant selection, it's actually pretty common in nature. So it seems antagonist selection is not very present, or we didn't detect it often in nature. And something I like to study is not looking to a monomorphic trait, it's looking at multivariate analysis by taking account not only one trait, but all the genetic architecture, so the genetic trade-off between multiple traits, and thus across the sex. So for that, we're going to expand the RFM, so this cross-sex genetic correlation, into the B matrices per se. I'm going to explain what it is after. And for that, we need to estimate the GFM, which is the sex-specific genetic matrices. This matrices, which is a giant one, is composed of the GF, so the genetic matrix for the female, GM, the genetic matrix for the male, and the B matrices, which are very interesting, which is the between-sex genetic correlation matrices. And in this diagonal, this is where we have the estimation of the cross-sex genetic correlation for each of the traits we're interested in. And the fundamental question in my science, in general, is how these B matrices and how the cross-sex genetic correlation can actually affect the sexual dimorphism evolution per se. So is that pretty rare? Usually I do a lot of laboratory, but this time I decided to do something more easy. I collected the GFM on the literature, and so far I found only 24 papers who estimate the GFM. We have 20 complete sex-specific genetic matrices, 29 B matrices in the data set, and just to show a bit like a statistical description, most of the GFM have been estimated in vertebrates and Drosophila, unsurprisingly. Mostly in morphology, but we have evidence in behavior, physiology, transcriptomic, and also life history. The method used is mostly frequentist approaches, but Bayesian has become stronger now. And unsurprisingly, these matrices have been estimated in laboratory environments in general. And so what I did with these new data sets, first of all, I tried to kind of investigate the parameter of the B matrices with these different components. So in the diagonal of the matrix, we have the shared genetic variance, so the cross-sex genetic correlation. In the up diagonal, we have the cross-sex, cross-trait genetic covariation, and something which is very particular of this B matrices, it's asymmetrical. So the upper, so we can see we have different colors, so the upper triangular and lower triangular are different. And I did a lot of simulation looking at the effect of the B component on the predictor and response selection, but I don't have time to talk about that today. I'm going to talk about something a bit more exciting for me, which is kind of a new approach to kind of investigate the antagonist and concordant selection approaches. So the B matrix, I talk about it, but what is about? So it represents the sex-specific pleiotropy loci underlying multiple traits, and the asymmetry, which is kind of what I'm focusing on this talk, it originates from this disequilibrium on this loci, which should occur during the process of genetic dissociation between the male and female. So if GF and GMM are different, we expect the B matrix to be very asymmetrical. When I look at my data set, only 5 papers have estimated the asymmetry, so it's a component people don't take into account often. And when we talk about the asymmetry, I will just point these 2 papers in evolution. The first one shows the B asymmetry evolve along the population adaptation, so it's important for the evolution per se. And also, the second paper shows the B asymmetry influence the B matrix constraint, and also produce sex-specific difference in the response section, delta Z. And something I want to point, it's all these 2 papers, it's only one population-based evidence. So that the idea of this collecting data set is to have more B matrices to explore a bit more how these sex-specific matrices can interact with selection per se. So when we look at the range of asymmetry I have in my data set, it's pretty big, so I'm pretty happy. I have like some variation to play with. And when we look at the difference between species or trait of interest, like physiology, behavior, we didn't find any difference on the B asymmetry per se. When we look at the correlation between the B asymmetry and the similarity between GF and GM, what we find is a negative correlation. So what does that mean? I mean, so each point here, it's like a matrices per se. Can I have a pointer?
Yeah, just drag the mouse over to your right.
Oh. People see?
To your right.
But it's fine. I'm going to try with my finger for far away with the force. So each point it's a matrix per se, and we can see clearly when the GF and GM are very similar, we have a very low asymmetry of these B matrices. Okay? And the kind of like turnover I'm doing here, it's what does that mean for me? That means the B asymmetry of a confirmation is associated to the genetic dissociation. So more the GF and GM are different, more we have a pronounced asymmetry. Okay? And due to the fact that GF and GM similarity is kind of a proxy for the shared genetic variance per se, we can say just by this correlation showing actually the asymmetry have an opposite effect than the shared genetic variance. Okay? So the first point of this talk, I show a new data set, which is kind of interesting for people who want to work on it. I show these B matrices associated to the genetic dissociation between the sex, so should facilitate the sexual dimorphism. Already? Oh my god. Okay, let's go. So let's talk about these other approaches. And I'm going to show you why I find that interesting. First of all, we take these matrices, and what we can do, we can rotate the axes using orthogonal vector, producing a new genetic matrices. And the idea is like when the male and the female are oriented in the same direction, you have a concordance axis which is created, and when the male and the female are on opposite direction, we have actually antagonist axis, okay? And interestingly, the GFM have 3 matrices, and the GCA, we also have 3 matrices. We have the concordant genetic matrices, which shows the genetic variance which is associated to the response to the same direction for the male and the female. We have the antagonistic one, so the orange one. So we show the genetic variants that allow a response in opposite direction for male and female. And so when we look at the decomposition of these different matrices between the 2 different subspaces we have in concordant and antagonistic, we can see in general for all the matrices here, which are ordered by asymmetry, the letter are just the label for the paper I took the matrix, so we can see it's mostly gray. So most of the variance of the GFM actually is sexual concordant. So what does that mean in biology? That means the GFM should produce more monomorphic trait in general. And now I'm going to talk about the last matrices within this GCS, so the BCS, the between concordance and antagonist genetic space. Interestingly, for people who want to read more about these matrices, what it do is capture only part of the B matrix information, so it captures only the cross-sex, cross-trait genetic correlation and its asymmetry, excluding the shared genetic variance. So I think it's interesting because now we can explore a bit more this asymmetry between the genetic correlation. And something important about this matrix, I put indirect because actually it shows the genetic correlation leading to indirect response to another subspace. So we have the effect of concordance in the production of dimorphism and the effect of antagonistic producing monomorphic trait. And so now we have this idea of direct and indirect response. How we can dissociate them? We can project a vector of selection to estimate the response to selection, but I think the facilitated way to do it is by just measuring the size of the matrix per se and compare the size of the BCA compared to the GA or the GC per se. And so the idea is if BCA is lower than GA, that would mean we have more direct response. If the BCA is bigger than the direct matrices, that means we have more indirect response to produce this new trait. And we can just do a log ratio of these 2 matrices, just to show you when we have positive values, I mean we have indirect response, that means the BCA is bigger. When we have a negative value, we have more a direct response. So the BCA is lower than the GA. When we look at all the matrices and we look at the difference between BCA and the concordance genetic matrices, we find all the values have negative. So what does that mean? That means in general, monomorphic traits are produced directly by the genetic concordance selection. So more direct response in concordance space. And when we compare the BCA with the other matrices, this is very interesting, I think so. It's first of all, we see a lot of variation between the size of the matrices. But in general, what we see is BCA, most of the time, is bigger than the GA. So that means we have the indirect effect from the concordance space into the antagonistic space. So in general, what does that mean in sexual dimorphism? I will say the sexual might be produced mainly by indirect effect of the concordance selection. And that's pretty interesting, I think so, because it's very counterintuitive when you think about it. And that's kind of what I want to show you guys. So I produced this new data set, I think it's very interesting, and if people are interested to collaborate with me, it's kind of cool. I show the B asymmetry is very important and actually facilitates the sexual dimorphism. I also show by using these new approaches, we can be implied for other type of analysis, like juvenile versus adult, different group, or etc. We can show for the GFM per se, we show more sexual concordant genetic variance in these matrices, and also the dimorphism in most of the case is produced indirectly from that one space to another one. So if I go back to the initial point of the talk, which is the classic antagonist selection framework or scenario, using multivariate approaches, I can show the B matrices actually capture constraint, but also enhancement of the sexual dimorphism. So I think it's important to have a bigger point of view of multivariate, so we can actually estimate better how sexual dimorphism evolved by looking at multiple traits. And also, I show more evidence for nuancing the antagonist selection scenario. And I think in the future, we should look more into concurrent selection and asymmetry between the sex to study the sexual dimorphism per se. Voilà. This is what I want to talk. I just want to say thank you to my team and also for Jacqueline because she helped me a lot about the mathematics. And also thank you for listening to Quantitative Genetic Talk. And if you have questions, you have my website, my email, and I'm very enjoy quantitative genetic and very happy in general to talk about mathematics. So thank you.
Hello everyone. Thanks for sticking here to the end of the day. And thanks, of course, for the invitation. I'll try and keep it short and so that you can go and have your dinners. Let me show the screen. Yeah, I thought I'd tell you about some of the work that Luke Holman, who's there, of course, and Tim Canallan and I have been doing to look at sex differential selection in the UK Biobank. Of course, sex differences in selection being sort of a type of trade-off, so I thought it would fit quite well with this symposium. And to set this up, I mean, one of the nice things I think to myself about being an evolutionary biologist is that we have a century or so of theory about various kinds of adaptation, including sex differences in selection and so it's the evolution of sexual dimorphism, but also all sorts of adaptation, mating systems, sex chromosomes, and so on. And one of the nice things also about being an evolutionary biologist these days is that we have access to large samples of genome sequences and also measurements of traits and fitness from various populations, and this gives us the hope, at least, that we can test some of these long-standing theories about adaptation. And in this case, the focus will be on the evolution of sexual dimorphism. Now, when it comes to studying sexual dimorphism and its evolution, we sort of have a bit of a challenge trying to use these new techniques, which is that say if you're trying to study local adaptation, well, you can look at two populations and they've genetically diverged to some extent, and you can do analyses that look for differentiation, and this can tell you something about the extent of spatially varying selection. But in the cases of males and females, when those are your two populations and you want to infer sex differential selection, males and females are part of the same population. So every generation, they come together and any allele frequency differences between the sexes are erased. And so this poses a particularly unique challenge for actually finding signatures of this selection in the genome. And this is the challenge that we wanted to try and overcome to some extent with Tim and Luke. And so that will be my story for today. And so this we chose to do in humans. Not everyone's first choice of organism, but for doing this. But humans have a number of advantages in this context. One is that you can have very large sample sizes of individuals, and as I'll describe, some of these methods to detect sex differential selection have relatively low power, and so it's particularly good to have very large sample sizes when it comes to this. Of course, we have large samples of genotypic information, and in the UK Biobank, we also have fitness estimates. In this case, the reported number of offspring that males and females of this population have. And so we use this information to try and test various theories about sex differential selection. So our broad approach to how to actually identify sites under sex differential selection was as follows. You have a sample of females at conception, so these are zygotes, and a sample of males at conception. And at conception, they have identical or essentially identical allele frequencies. So there's white alleles and black alleles here in this particular cartoon. But then you can imagine that maybe alleles have some effect on viability, and maybe this effect on viability is different between the two sexes. And so here the white allele, for example, confers some protective effect on viability in females, and so it's enriched in the sample of female adults. And maybe the black allele has some protective effect on mortality in males, and it's enriched in the sample of male adults. And so you can see that the allele frequency differences between the samples of the two sexes tell you something in principle about sex differences in viability selection. And the same is true if you have information on reproductive success of each individual. You can imagine that there's a sample of gametes which depends to some extent on whether alleles have a positive effect on your reproductive success or a negative effect on your reproductive success. So for example, the white allele might allow females to have more children for some reason, and so it would be enriched in the sample of gametes, and then the black allele might do something similar in the sample of males, and that might be enriched in the sample of sperm. And so you have this situation where allele frequency differences between adults tell you something about viability selection between the sexes. In the gametes, they tell you something about viability and reproductive success, and you can also sort of take the difference between the two to just look at reproductive success. So you can look in this way at if you have adults and if you have their information on their fitness, you can partition sex differential selection of a whole life cycle and use these FST, which is a measure of differentiation, to quantify this selection. And so with this, we wanted to answer 3 questions. I've sort of gone for breadth over depth here, so please feel free to ask me methodological questions later if you want. First question is this selection actually present in the genome? Can we detect it? And we should be able to, because of course we find examples of sex differential selection on traits plentifully. And so here you can see these plots, which I'll use quite a lot. So the distribution of FSTs is split into quantiles. And so each point here represents, each category here represents a particular quantile of FST, and then the y-axis is the percentage of sites that fall into each quantile of this null distribution. And so if there's no selection at all, then 1% of sites should fall into each of these 100 quantiles. And that's what you can see if you randomly assign males and females to be any random individual, if you permute the data, in other words. But if you look at the actual data, there's an enrichment of sites with high FST, which is this indicative of sex differential viability selection. And of course, we're not confident that any one single site is under selection. These methods in general have low power, but we are confident that as a polygenic signal, it's there and it's genuine. And this is also true if you look at reproductive success and if you look at total selection. There is always this enrichment in the upper tails of the null distribution of FST. And yeah, and the other thing that's nice is that these sites that have high FST also tend to be found in coding regions and tend to have some associations with traits. And so this gives us some reassurance that they're not just artifacts, but they have some genuine phenotypic effects and therefore are more likely to have fitness effects as well. So yes, to answer this first question, can we detect it? Yes, we can certainly find polygenic signals. A second question we wanted to address is, are these sites more common in the autosomes or the X chromosome? If you're in this field, this is quite a common source of disagreement. Some people think it's more likely to be on the autosomes, others more likely on the X. So we just wanted to go into our data sets and compare X and autosomes. And so here you can see the X chromosome, same kind of plot, enrichment in the upper tails on both. The question is there more of an enrichment than what you would expect on the null, say on the X chromosome? And so here we developed a null distribution. So we assumed that there was a random distribution of sex differentially selected sites across the genome. And then we, from this null distribution of sexually antagonistic sites, we computed what the expected FST enrichment would be under this null. And then we compared our data to this. And so what you can see actually, these are bootstrap estimates of the X to autosome enrichment, is that there's no difference between what we observe here and the null if there's a random distribution on the autosomes of the X. And this null is not at 1-to-1 because the X chromosome is haploid, and so that enhances signals of FST. But in any case, this is sort of a slightly anticlimactic result maybe, but it was a nice, we wanted to find an unbiased test of this hypothesis, and this is as close as we could come up to, and there doesn't seem to be a particular enrichment either on the X or the autosomes, based on our analysis. And finally, we wanted to ask what are the evolutionary, what are the modes of evolution that affect these sites that are putatively under sex differential selection? Are they just evolving under drift because these are humans, small population sizes? Are they under purifying selection? I mean, there's sex differential selection, but maybe in general the ultimate fate of these alleles is to be lost. Or are they under balancing selection? And one way we tested this is to look at the correlation between FST, these measures of selection, and the minor allele frequency. And what you can see here is that sites that have high FST, so are more likely to be under selection, also tend to have high minor allele frequencies. They tend to be more common in the population. And this should actually this was very surprising to me, and still is somewhat surprising to me, because if you look at any loci that affect quantitative traits, loci with large effects tend to be rarer. And here we really find that they're systematically more common than sites with low FST. And this is true whatever the component of fitness that you look at, so whether it's viability, whether it's reproductive success, or total fitness. And this is really incompatible with the model of mutation selection drift balance, because you would expect that sites with larger FST and therefore larger fitness effects across the sexes would tend to have low minor allele frequencies, but this is not what we find at all. So certainly it's not purifying selection, or not pure purifying selection. It's also not long-term balancing selection. We looked at the correlation between FST and allele ages, and what we found is that there's no substantial signal. There's a little bit of a signal for reproductive success, but in general, there's very little indication that sites with high FST tend to be older than you would expect. And yeah, and so this doesn't, well, it narrows it down a little bit in the sense that it's not long-term balancing selection, it's not mutation selection drift balance. And so these sex differentially selected sites are potentially evolving under drift, which would be very plausible in humans given the effective population sizes. Or they could be evolving under short-term balancing selection. Yeah, and with that I would like to thank funding and of course Tim and Luke for this really pleasant collaboration, and you for listening. I'm happy to take any questions.
I would take questions, but I think I think we could go on to right. It's the end of the session, right? It's 5:30. So I would just like to thank everyone for coming to the session and all of the speakers. I'd like to give a round of applause to everyone who spoke in what's been a wonderful symposium. Thank you. Thank you, Carl.
Actually, I think there's one more thing, which is actually you're better positioned to say this than me, but I'm just reminding you out loud, which is that there are a lot of unknowns and kind of broad inconsistencies in this field with different kinds of selection over different contexts, and so that's why we wanted to bring these different factions of people together in the same room to get the same thoughts going among these different sub-disciplines that don't usually talk, and so there might be scope for some type of special issue or idea, or potentially some kind of review paper. So if you've got ideas about that, come talk to Luke, maybe?
Oh, yeah, either of us, yeah. I should also say I'm the commissioning editor for the Journal of Evolutionary Biology, and part of my role for that is to commission special content for the journal, things like reviews, commentaries, perspective, and even special issues. And so where people have an interest in coming together, for example, discussing the commonalities and differences between various things that we've talked about today,
for example, sporophytes and gametophytes, different morphs of roughs, males and females in many of the talks, social insect, queens and workers, and many more. I think there's something there. I think an emerging theme of some of the talks is to what extent is balancing selection a thing? When selection operates in this way, we've had two countermanding viewpoints on that at least. A lot of people looking for evidence of balancing selection in various interesting ways. There's methodological aspects of it. I'm just really wary of leading such an endeavor at the moment with my other commitments, but I'd be super interested in in talking to people that have ideas for papers, whether as a group effort related to the symposium or just your own ideas with your own buds that you want to write with. Thank you for reminding me, Carl, and just, yeah. I think that we really achieved what we set out to do, to bring together like absolutely fascinating collection of talks on disparate but related things. And I've been really pleased with how it's worked out. And so thank you all for the engaging questions and talks today. I'll see you all later. We're here for several more days. Let's talk to each other more. Thank you.